So today we're going to continue our march to understand a bunch of different bioorganic polymers. We saw that amino acids make up proteins, and of course we've already talked about the fact that monosaccharides make up polysaccharides and complex sugars and stuff like that. And so we also probably won't have much time to talk about nucleic acids, which are the monomers that make up DNA and RNA. And we probably won't talk at all about lipids, which are usually made up of glycerol and fatty acids, or can also be made up of a bunch of different cholesterol type isoprenes. So what I'm trying to say is that we're just getting a real kind of cursory view towards a lot of this stuff that you'll really start to develop as you start to take more biology and more biochemistry. So again, I have all these slides posted. We're just going to kind of run through what I think are probably the most important concepts having to do with amino acid reactions. So probably the most important thing to remind yourself of is that we've got carboxylic acids, right, that have a pKa right around 5, and we're actually going to be looking at the pKa of 10 for this amino group right here, or protonated amine, ammonium, I guess I should say. And so what we're going to really work towards is understanding the zwitterion formation of these things, right? This idea that if you're given some sort of pH, you're going to be able to tell whether or not you're in just a zwitterionic form, or by the way, zwitterion means like literally two ions, right? Opposite charge in the same thing, kind of similar to that phosphorus ylide we talked about, but the difference is a zwitterion can have a positive and negative charge literally anywhere, whereas an ylide has to have them right next to each other. So we're going to keep in mind that most amino acids, even though we generally write them like this, in biological systems, because the pH is very close to 7, we actually have the deprotonated version of the carboxylic acid group, and then the protonated version of the amine here. So knowledge of those two pKa's is going to allow you to have a pretty good idea of what's called the isoelectric point of different amino acids. This is the simplest amino acid called glycine. And yeah, so what you can see is that if you have some sort of pH that is, let's say, very near your isoelectric point, right? You're going to have something like you can see, and for this particular one that ranges from 4 to 8 pH units, that you actually exist in what's called the isoelectric form for glycine, right? In other words, where the plus and the minus charges are perfectly balanced. Obviously, if we put some sort of R group on this thing that itself has some sort of carboxylic acid or amine or some other acid-base active type thing, that is going to actually change the isoelectric point, right? Because that point where every positive and negative charge is perfectly balanced is going to change a little bit. And this is kind of my long-winded way of kind of getting to this idea in the first couple of weeks of biochemistry, that you're able to, based on your knowledge of pKa's from organic chemistry, decide where protons are, right? Whether or not something is protonated or not, depending on a pH. So even though you'll talk a ton about isoelectric points and why they're important, I always kind of bring it back to organic chemistry, where my knowledge, where I know that the general pH of this thing, the general pKa of this thing is right around 5. That tells me that if this thing is going to be protonated, it is going to be protonated at pHs that are like 4 and below, right? That's where we're going to have a complete excess of H plus ions, so we're always going to have the protonated form, right? So I kind of imagine that pKa number is kind of telling me plus or minus 1 pH unit where I'm going to have a species that's either completely protonated, where we have a pH that is lower than the pKa by 1 unit, or completely deprotonated, where there's a pH 1 unit above the pKa, and then in between there's going to be this buffer region, which is highlighted right here, right? Between those two values, where you're going to have maximum buffer capability, because that's where you're going to have a little bit of the carboxylate and a little bit of the carboxylic acid. Same kind of thing can happen up here. Give or take, this thing has a pKa, this thing has a pKa right around 10. So what that means is that if we have some sort of pH of a system that is 1 unit below that, that means it's going to be primarily in its protonated form. And then if it's something with a pH that is 1 unit above that, so in this case 11, this thing is going to exist in the completely deprotonated form. So kind of putting that together is going to allow you to really determine where your protons are in a protein, and that turns out to be a pretty important thing. So as a general rule of thumb, even though I consider these isoelectric points not super helpful, because really what I'm interested in is an organic chemistry, as an organic chemist or even a protein chemist, is actually knowing where my protons are in terms of these amino acids. But this isoelectric point is definitely something you'll talk a lot about in biochemistry. And so you've probably remember from the introduction to amino acids that certain things are considered acidic amino acids. So these are going to have isoelectric points right around 3. And then you, of course, have basic amino acids whose isoelectric points are around a pH of 9. And then everything else kind of runs in the middle. So this might be helpful just to keep track of what actually happens in an acidic solution. For some reason, the fact that acidic solutions have a lower pH sometimes confuse students. So that's something that's really going to help you out in biochemistry if you just kind of can stick to that idea that the lower pH means there's more H pluses present, right? And so you're going to, if you're 1 unit or more below the pKa of something, that means that you're going to have everything protonated. And the opposite is, of course, true if you're 1 unit above that pKa. In terms of reactions that these amines undergo, or I should say reactions to produce amino acids, pretty much all of these methods are going to depend on kind of a modification of the reductive amination that we've already talked about, right? This idea that we're going to treat something with some sort of amination and then we're going to simply reduce whatever the product of that is. Your text makes a big kind of stint about this idea that it's biomimetic. And really, the biosynthesis of amino acids is pretty straightforward in terms of like an organic context. But when you really start to look like at biochemically what's going on, one of the themes of kind of my understanding for biochemistry that I hope that you're going to get to at some point is to recognize that all these kind of like more simple looking things that we learned in organic just have a certain more complex analog in biochemistry. So a really great example of that is in when we actually make amino acids in the body, right? In this particular case, we can make it from one of these Krebs cycle intermediates here, which you'll learn about eventually. But you'll notice that like the reduction, right, which we saw in the previous thing, which we did with H2 and palladium, obviously we don't have H2 palladium in the body, but what's going to happen is your body's going to have the analog for that, right? And so this thing, nicotinamide adenine diphosphate, which has this like gigantic structure if you really look at it, right? There's a ribose sugar down here and there's all kinds of weird functional groups, but really all we're doing is like transferring that H, right, as a source of hydride, right? It really is just a big giant H minus when you really look at it. And then as a result of that, right, you transfer the H minus and you end up with NAD plus, right? And so there's all kinds of different reductions and oxidations in the body that involve things like NADH, NADHP, which is like a phosphorylated version that is usually saved for pathways where you need to do some sort of oxidative reduction, oxidation or reduction that involves like the synthesis of something, whereas your body will typically use NADH, the non-phosphorylated version, in the metabolic processes and things like that. So there's all kinds of stuff to learn, but the important thing to realize is that all these complexities really do serve like a pretty simple purpose. By the way, there's other pathways where you use a type of flavin dinucleotide, but really they're all doing the same thing. They're just, different pathways perhaps that require certain different structures, right? And different enzymes require different things. And biochemically, eventually you get to the point where you realize that you can kind of transfer amine groups from one thing to another. So pretty similar to what we saw before in terms of the kind of dicarboxylic acid. It turns out that your body has these great abilities to use some sort of amino acid to transfer the amine from one thing to another. So this is just something that you'll spend a lot of time talking about. I don't know that it's important enough to make a note card, but a lot of the biological processes for producing amino acids that are kind of truly synthetic in nature, these things are not as important anymore because, quite honestly, we've learned ways to kind of design biological systems that'll do a lot of this stuff for us. But back in the day, there were, of course, a bunch of dead white guys that came up with some pretty fancy ways of making amino acids. This is kind of a combination of the Gabriel reaction that we talked about already in amine chemistry, and then also the malonic ester synthesis. And this is really just a combination of the two of those things. You're basically just doing malonic acid ester synthesis type chemistry appended to this big giant thalidomidmalonic ester. And then eventually through hydrolysis, you kind of cut that sucker off. So this is kind of it's not a terribly common thing to see anymore. Slightly more common is going to be something called the Strecker synthesis, which is a type of reductive amination that we didn't talk about specifically, where you actually are going to have an amine that you form an imine when you attack the aldehyde, and then you in situ we're going to attack that thing with cyanide, and after hydrolysis you end up with any kind of amino acid that you'd like. Of course, this isn't an enantiopure reaction, but now they've figured out ways to do this kind of thing in an enantiopure fashion so that you can get just the L-alanine, or I guess if you want to, the D-alanine. As I said, back in the day, this thing was a lot more important. This is something that I've seen in different contexts for making what's called non-natural amino acids, right? Where we have R groups that maybe instead of being an alanine a methyl group that makes this thing an alanine, just unusual things that don't actually happen in nature. So it's probably a good thing to talk about in a little bit more detail. Your book has a really good mechanism for it, which is not terribly interesting, I don't think, but it's sometimes referred to in certain biological contexts. Your text goes through a couple of other semi-interesting reactions that amino acids undergo. Honestly, they're not super common. Things like the different ways to sterify the carboxyl group, and then there's a little bit of a section on selective deacylation in order to get a particular race mate. So you may want to look at that. The reaction with ninhydrin here, I only highlight because it's a really cool demo to do, and also pretty important for forensic chemistry. Turns out amino acids really like to react with this ninhydrin, and it makes this like UV active compound that a lot of forensic scientists use to be able to visualize like different amino acids left behind in crime scene. It's also important occasionally in like a biochemical lab where we're trying to maybe label some sort of chromatography with a stain. By far the most important reaction for amino acids is in its formation of an amide bond between different amino acids involved in a peptide or and speaking more largely into a protein. One thing that's really interesting is that one of the things that makes this bond so incredibly strong is this fact that we've got resonance stabilization here that ends up with what kind of looks like a double bond between the carbon and the nitrogen, right? And so even though this thing in an amide is always written as a single bond, it's actually got quite a bit of double bond character. And this in biochemistry will lead to all kinds of interesting looks at the three-dimensionality of proteins and things like that. In fact, like the alpha helix and things like that, that you'll talk a bunch about in terms of the secondary structure of proteins, has to do with that. So not surprisingly, if we have the right conditions and we bring two zwitterionic amino acids together, we do end up with this thing, which in biochemistry is called a peptide bond, and an organic chemist perspective, that's called an amide bond. And this, of course, liberates water. This is why just about all of the bioorganic polymers link together by something called dehydration synthesis, right? The loss of water, ultimately, even though when you look at the actual biological production of these things, it doesn't necessarily just happen as simply as losing H2O. There's a lot more complexity there. Overall, that's what's going to happen. So you might have heard me use the word polypeptide. Usually I reserve that for, let's say, a polypeptide usually isn't any greater than 10 amino acids. They actually define it in terms of molecular weight. Nobody really does that. And protein, in terms of me talking about something as a protein, it's usually something that is certainly greater than 10 amino acids linked together. And honestly, many of them have hundreds of amino acids. Another thing which you should probably know, so here's just a pretty simple proteinaceous hormone here called bradykinin. You'll notice that this N terminus and the C terminus becomes important in terms of giving what's called the primary structure of proteins. So these little three-letter abbreviations are something that you're going to probably have to memorize. You'll definitely in biochem have to memorize all 20 of the naturally occurring amino acids. And I know in some of the classes, not in mine, but in some of my compatriots classes, they had to memorize the different one-letter abbreviations, which are a little bit different. So for instance, I think arginine is probably an R, and proline is a P, and glycine's a G, but then you have I think it might have been an F for phenylalanine here. But the important thing to recognize is that in terms of giving that primary sequence for a protein, is you always go from the N terminus to the C terminus, whether or not you use the three-letter system or just the single-letter system. When you start to talk about what's called tertiary structure, which is kind of like the boogery shape of proteins, there are other important reactions that you'll want to be aware of. One is this idea that thiol groups, I think we've even talked a little bit about the importance of thiol chemistry to a biochemical system. Here's like a great example of something that's really important. When you have cysteine residues, those things can form what's called the disulfide bridge, which is really a protein's way of really holding on to its structure.